Good afternoon, everyone. Happy to have you here with us today, and I'm excited to be here with you for the ASGCT Lunch and Learn series. I want to thank the ASGCT Patient Outreach Committee for hosting, I think, an important discussion today. I'm Laura Hamid, and I'm the Executive Director of the Columbus Children's Foundation. And today we're going to discuss how AAV vectors are made. At CCF, we focus on the acceleration of the most effective gene therapy treatments for patients with ultra rare diseases. And in many of our conversations, we continually hear that manufacturing remains one of the biggest hurdles and the biggest mysteries in advancing programs. What is AAV? Why is it so complex? Why is it so expensive? Why is it so challenging? And what are the issues we are seeing with this modality? And I'm really thrilled to have with us here today two experts who will help us answer these questions. We have Michelle Berg, who's the president, GMP nucleic acids at Aldevra, and Dr. David Dismuke, who's the chief technical officer at Forge Biologics. Michelle and David will do a presentation to help unravel these mysteries around what we need to know when we consider AAV gene therapy manufacturing. Please add questions into the Q&A section and along the way. And after the presentation, we should have time to address a number of those questions. So with that, again, thank you for being here. Stay tuned to an amazing presentation. And I'm going to hand it off to David and Michelle.
Great. Thank you. So let me get things started here. Hopefully everyone can see the screen and get into the slideshow. So again, it's our honor to be here and to present this. It's obviously very pertinent to the community out there of patients and advocates about the vectors that they use in these trials and how they're made. And with that, I'm going to hand it off to Michelle to get us started.
Thanks, David. Thanks, Laura. Also, thank you to ASGCT for putting this curriculum together, the opportunity to share. Really excited to do so with a longtime colleague and friend in David, who most importantly is a top expert in AAV biology and manufacturing. And want to point out that we have tailored our talk today specifically for an audience of patient advocates. And so our objectives are, of course, to share about AAV, why we're excited about it, dive a little bit deeper into the challenges that you hear about. And most importantly, we hope to empower you patient advocates with more knowledge as you navigate your specific disease journey. So David, you can jump to the next slide, where just want to call out that the content has been intentionally curated and developed to build upon one another. So if you did miss the previous lunch and learns, I think it's of value to go back and watch Gene Therapy 101, as well as the intro to viral vectors. So you can get the next slide there, David. Thank you. All right, so what is AAV? AAV, or adeno-associated virus, was discovered first in 1965. And over time, it's become understood that it has some pretty interesting properties that we'll share more about. This virus-like particle is not able to infect or replicate inside a host cell without help from another virus on its own. And instead, I'll point out that it does not have an envelope like other viruses do, but rather an aggregate of proteins that form a shell or capsid. And you'll hear more from Dr. Dismuke about capsids and really what their importance can play. An additional property that is important is that the DNA carried by AAV can be engineered or altered, and that's something that we're taking full advantage of in gene therapy. And so if you look at that first photo on the bottom left, that is one of the three viral proteins that come together, and there are 60 of these subunits that form that outside shell or that capsid that you see in the middle photo. And that's important in protecting the genetic payload and importantly, the genetic payload that we want it to protect and carry forth that is found inside of the capsid. And that is the last photo that's on that bottom right.
Yeah, and just point out the sort of overall complexity. Maybe you can see that a little bit within where the purple and the teal and the green come together. How the capsid molecules, those BP viral proteins, come together to create this lattice, and they're really intertwined to each other. So how they form is relatively complex, which is I think pertinent for this discussion of why it's difficult to make.
Thanks. All right, you can go to the next slide. So won't go through this exhaustively, but point out some of the highlights that since its discovery in 1965, there's been a lot of effort to just understand the basic biology about AAV, how it works, and really growing in the understanding of how it can function as a delivery vehicle for gene therapy. And a lot of time has been spent in improving upon the design for the purposes of being safe, safety, and then of course, how we can utilize it deliberately and specifically for delivering for clinical trial use to result in efficacy or showing results in humans. And so in 2008, that was the first time that actually was demonstrated, and resulting in eventually the 2012 first approval of an AAV delivered gene therapy by the EMA in Europe, and excitingly the first FDA approval in the United States by the FDA followed in 2017. And in preparing for this and discussing this, it's really important, we felt, to point out that a lot of the knowledge, a lot of the progress has taken place over this last decade, and that knowledge is continuing to grow. We'll speak more to that later in the presentation. But we wanted to take this opportunity as well to underscore that throughout this 50+ years of working with AAV, understanding it and utilizing it to deliver, this hasn't just been an endeavor taken on by the academic and scientific community. That really much of this effort over the years has been arm in arm, side by side in the trenches with advocates, representing caregivers, representing patients affected by certain genetic diseases. And so this field has much to owe to the patients, to the patient advocates as a result of that effort. So David, you can jump to the next slide. So I won't again exhaustively walk through this because we've mentioned, I've mentioned much of this already, that in researching and understanding AAV, we've shown through many different animal studies and certainly in clinical studies in humans that there is a strong safety profile for AAV. And that importantly, it is not something that can integrate into the host DNA. And that's important because that does not, that reduces the chances for causing cancer. And again, you'll hear more from David on how this is utilized to engineer and deliver the desired genetic payload, but also how we can, there are multiple opportunities and methods, that is for generating and purifying AAV that also demonstrates a really nice stability, meaning that it can stay on the shelf for quite some time and maintain the integrity and maintain its ability to do what it's needed to do over time. And so some of the, I guess, drawbacks that we would call out, you might have heard this before, but there is a limitation on what you can put into an AAV with regard to the size of gene or the size of DNA that can be delivered. And so I think the takeaway that I'd love to leave here is that AAV, while we're very excited about it, is not for every single disease, is not going to be able to deliver what's needed, for every disease. And that's where we have to be very thoughtful about how that's applied and the disease types that we think about utilizing gene therapy delivered by AAV for. And so this is just demonstrating that, again, as we're excited about it, so is the field. AAV far outweighs the viral vector delivery of choice from any others. So 46% currently in the clinic are using AAV to deliver. And again, pointing out that we have 2 currently marketed and approved commercial products that use AAV in Luxturna and Zolgensma.
So, of course, you just want to get into the complexity of AAV, and it's obviously great that we're able to produce these vectors, and they've been approved for clinical trial or for approved products, and many of them are in the clinic. But they're more complex than most drugs, and this is really talking about biologics as a whole are more complex than small molecules. And we think about drug discovery. And we think about manufacturing of any drug, how complex and how hard is it to make. So here is just an illustration of the size differences between something very typical like aspirin of 180 Daltons, and comparing that to something like insulin or erythropoietin or a monoclonal antibody. And so you can see really in the size difference and the little blobs there that represent sort of a molecular structure, where it gets more and more complex as you get larger. And we're also getting into this a little bit, but where aspirin or other small molecules are synthesized, we utilize nature to produce biologics that are produced in living systems. And as such, we say this all the time in manufacturing, but the process is the product, and so there can be variabilities that don't exist really within as much within small molecules. And so you compare sort of these sizes and think about the heterogeneity that you would see within some of these biologics, and you compare that then to AAV, which is 25 times larger than even a monoclonal antibody. It really takes complexity to a different level. And when you also factor in, again, as we looked at the structure of AAV, where these subunits come together and are really interwoven into each other, again, also adds to the complexity. And then on top of that, you're adding not just a protein shell, but you're also adding the nucleic acid that is packaged into that particle for delivery. And we have some differences in what those can be, whether they're single-stranded, whether they have some ability to recombine immediately to form a self-complementary or double-stranded structure. But they become very complex. But we're able to use the vectors here, and they're complex. Also from the fact that how they interact with the cell and how we then also test them. So thinking about that, I put together this table and it's a bit of an eye chart, I apologize, but it really is to demonstrate the differences of complexity around the synthesis, whether it's for looking at a standard biologic versus a viral vector, or even a cell therapy, and understanding the complexity when it comes to synthesis or the structure or the cell type used. So just kind of walk through these really briefly, but when you think about AAV is produced in a cell type that's usually eukaryotic, so it's a human or like an insect cell, versus something like an E. coli or a yeast that is produced for a lot of biologics, versus something where it's synthesized from chemical structures. And so because of that, the structure overall is more complex, and it's not as understood as a small molecule. And we think about small molecule structure, if you make another small molecule in a slightly different system, they really become still identical. Where if you make an AAV or another biologic within a slightly different system, they're similar, but they're not identical. And so it's a really important point to think about the differences that we might introduce within making any biologic as we change the system slightly. And then there's also just increased risk around how we sterilize the products. For small molecules, you can actually heat them to usually high temperatures and terminally sterilize what's in the vial as it's in the vial, versus doing sterile filtration for a biologic, And some viral vectors, like some larger viral vectors, are actually difficult to filter. Luckily, AAV it goes through the sterile filters fairly well, and so that's not a problem. But if you compare that, again, this is maybe out of the scope of this, but cell therapies are too large to be filtered. So, and then we think about the scale of manufacturing and what is able to be produced from a single batch. There's you can produce a lot of small molecules at very large scale, and even some biologics at 20,000 liters, where typically the largest scale that you can get to for viral vector manufacturing is a 2,000 liter, and that may only produce 100 patient doses per batch versus 10,000 or hundreds of thousands of doses per batch. So I think it's just important to understand those sort of limitations around the manufacturing and those caveats as we think about that. It's really down to the complexity of the viral vectors. We also have the complexity around different types of AAV. And so you may hear this a lot within using an AAV9 or an RH74. Different serotypes that we use, and these are really, really beneficial for patients because then we're able to really traffic different types of cell types, different tropisms within the individual. It's important to note, though, that we have seen that there are differences between species. So a lot of the early work is done in the field was in mouse and rat models, these murine models. And as we moved into patients, oftentimes those vectors, those serotypes didn't work very well. Specifically, most of the early work was done with AAV2. And when those were put into the patients, they didn't. The safety was typically fine, but the efficacy was poor. And it wasn't until 2008 when moved to an AAV8 vector that was then used for hemophilia. We really saw this ability to get good efficacy within a patient population, and then later on, with AAV9 for SMA, again seeing the ability to transduce the cells of types better and to be able to deliver the gene of interest to where we want it to go. So just important to understand those differences and how they maybe impact overall manufacturing. So here on the right, you'll see some cryo-EMs that were done by Mavis McKenna's lab at the University of Florida, and you might note that they look pretty similar. They're not that different, and so we can utilize a lot of that similarity, but the differences are enough that it really changes a lot of how we purify the viral vectors and how they're produced in the cell. And importantly, once we've made them, what is their stability? An AAV2 vector, for example, has a greater propensity to aggregate than something like AAV9. And so we have to be thoughtful about that when we're producing these vectors. And it also changes how that vector may interact with the vial that it's being stored in, or as well as the delivery device in which it's being delivered to the patient. So we have to really factor all those things in and understanding the differences around those vectors. Hand it back to Michelle.
Yeah, so back over to me. Yeah, thanks for that, David. So here's where we get into the why things take longer than everybody would like it to take. So first, I'll say that everything is done with the mandate of keeping patients safe, but also we have to follow the necessary steps to develop a drug or gene therapy product that are outlined here. So within each of these steps, preclinical, clinical, commercial, is a series of not only critical tasks, but important decisions that as one builds or discovers a new gene therapy candidate that have to be understood in order to really, again, safely deliver and have the necessary and desired result. So for example, once you move through that discovery phase where you've identified a candidate that you think is going to have some potential and promise, and move it through that preclinical research stage such that you're now ready to bring that forward into human study. It's really important that before you do that, that you take the time to understand and develop a process that is robust and repeatable and can withstand the scale up, something that we'll talk about in a little bit here as well. And that prior to moving into clinical trial, you have to start considering starting material and where you're going to get that and generating that material. And in parallel, work on important animal studies called toxicology biodistribution studies, where you're examining, is this material being delivered safe to the animals that are under study, and where is that material going throughout the body, or where is that being distributed? And an additional step that you'll want to be considering and is needed is assays that are utilized, making sure that they're qualified for the purpose that they're needed and can take you the distance through clinical trial and set you up successfully for commercial. Likewise, as you're moving through the different phases of clinical trial study, and preparing for that success to move into commercial and approved marketed product, that there are important and critical steps there as well, such as process and method validation, really looking at having the supply chain defined and outlined, because there is a requirement, of course, to be able to continue to supply drug should that become commercialized. And oftentimes that comes with having backup or secondary sources for materials in that supply chain. And all along this way, preclinical, clinical journey, there's documentation, important capture of information, design of studies, results from studies that are assembled into the BLA and prepared for filing with the regulatory agencies. So the purpose of highlighting these steps is really just to share, again, there's so much that takes place at each of these different steps that are critical to not only delivering safely, but delivering in a way that the regulatory bodies can understand and be able to allow or approve moving to those next stages. And so, and if you have any other comments, David, happy to pause. Okay, great. So likewise, as we mentioned, moving through that timeline, preclinical to clinical and into commercial, there's also a simultaneous scale that's going to be taking place on quality as well as quantity or volume of material that's needed. And so in those earlier stages as preclinical research grade material and the quality oversight are appropriate at that stage in order to be looking at studies again in animal models, cells that are preparing you for that next stage of moving into clinic, where you will need more material because you have more larger participants typically in the study that require more material, larger numbers of them as you progress through the different phases of clinical study. But because of the criticality of moving into human subjects or human participants in clinical study, the requirement for quality oversight that's required by the regulatory bodies increases as you scale through those different phases of study, along with the volumes needed. And certainly that escalates with regard to quality oversight as you enter and prepare for commercialization as well.
Yeah, and so going back to the scale up, and we think about the size of the bioreactors, as Michelle was showing there. I just want to highlight the importance again. If it's a factory within a factory, and so the bioreactors obviously reside within the clean room facilities within the factory overall. But then really the work is being done, the vector is being produced inside living cells. And so we're really leveraging nature to be able to provide this service for us, that's where the manufacturing is most efficient. And so when you think about, a single 500 liter bioreactor, typically, cells that we have that, are 5 million cells per milliliter of culture, and you calculate that out, we're 2.5 trillion cells in a bioreactor. And for AAV, each cell is producing, roughly 10,000 to 100,000 particles per cell. And so, that's really where the work is being done on that per cell basis. And we're very thoughtful about that and how we're treating the cells when they're in that bioreactor. And so, this sort of focusing in on this factory within a factory, what we really need to do for the cell, whether it's an HEK cell line or it's a SF9 cell line or others, it's being able to get the genetic material into the cell, again, the blueprints for instruction and for making copies of the viral vector. And so, listed here is really taking the DNA, that we often get from Aldebaran, and complexing that with reagents to get them into the cell. Now, this could be done again through a different sort of delivery mechanism, like a baculovirus vector, which I'll get to in a minute. But it's really getting the DNA into the cell and then getting that DNA into the nucleus of the cell. And once that occurs, then the protein production ensues, and then the capsid itself is formed. And so capsid for AAV, capsid assembly occurs first. And then after the capsid is assembled, the copies of the genetic material we're trying to package, we call that the transgene. It's replicated and then inserted into this already formed capsid shell. Once that occurs, then we're really getting a vector release by sort of the breakdown of the cells over time, or by mechanical or chemical lysis of those cells. So, and then we get release. And as you see here, maybe we get release of capsids that have genetic material in them. We call those full particles. And we also get empty particles. We also get other things like partial, vectors that have, because this is a biologic system, we do get some partial packaging as well. So it's a heterogeneity around the particles that are produced. And then we would need to actually purify what we want away from what we don't want. So, again, as I mentioned, there's different systems for AAV manufacturing. Now, this is probably the top sort of 4 categories that are used in transient transfection of HEK, and that's the most widely used overall. There's also baculovirus SF9, which is used by a number of parties. That uses an insect virus been engineered to deliver the gene to insect cells. And then there's herpes virus, which are again, that herpes virus itself is changed so that it has less chance to cause any problems, and that is used to deliver the genetic material to the HEKs for production, as well as producer cells, where it's a stable integration of the genomic information for the for the AAV particles themselves, as well as the transgene, so they can it's stably integrated into the cell. There's caveats to all of these, for cost of goods, for example, for HEK293, again, which is the most widely used, is the highest, but then the timeline is the shortest. So that's really, time to get to the patients is one of our major concerns. And so that's usually why these are, utilized, and it takes the least amount of effort to tweak and change the system. It's the most platform that we have. But that being said, there really is no standard platform as you would see within, say, monoclonal antibodies that uses a fairly consistent process independent of manufacture. So there's also the sort of the caveats around what's made. So HEKs typically produce the 10 to 20% full particles, where other systems maybe have more. It's like in baculovirus or in herpes virus system. And just sort of, again, back to the time around development, where HEK is relatively quick, but it could take several years, for example, for a producer cell system to be fully developed and make that GMP clinical relevant cell line. And we have to make the starting materials for these systems as well. And so for HEK293, one of the reasons why it's so quick is there's not a lot of optimization that is necessary to just get started with production. You make the plasmids, you introduce them into the HEK293 or HEK293T cells, and again, fairly widely used and able to be exploited. For SF9, you have to first make the starting materials of the plasmids and then make what we call backmids, which are just basically the genome of the baculovirus itself, and then introduce that into cells and make recombinant baculovirus expression vectors that are then themselves introduced into SF9 cells. So again, a lot more complicated, and they have to optimize each of those steps. And for a herpes virus system, again, you need recombinant herpes viruses first, and then introduce them to the cell. So HEK is a little bit simpler, easier to go through, where other systems take a little bit more time to develop, even though they have some maybe a lower cost of goods and a bit higher productivity. So from the actual production side, what we really think about are two big categories. One is the upstream process, which we're using the bioreactor. Again, the cells are growing, they're actively producing the viral vector, making AAV. So we scale up the cells. And then once the cells reach the production volume here, showing a 500-liter bioreactor, then we would introduce the genetic material, the plasmid DNA, to transient transfection, complexing that DNA to get it into the cells. We hold them for a period of time, usually a few days, and until the cells are starting to break open, and then we complete the process by lysing the cells and removing any extraneous DNA that we don't need through a nuclease step. And once we have that gamesh, that now we have to purify the AAV away from all the cellular debris and other things we don't want. And so, that's really through, a series of steps that are outlaid here, and they could be different depending on the manufacturer and the system that's established. But first, we typically go through a filtration process to do the bulk of the work, to just remove a lot of the large particulates that we don't want. And then maybe go through a filtration process that reduces the volume a good bit. If you're starting with 500 liters, 1,000 liters, that's a lot of material. And then taking that through a chromatography step. And I'll go through a little bit more detail of what affinity chromatography and ion exchange chromatography are. And then that goes through, again, changing the composition, the formulation that it's in, to what we want it to be for the delivery, and then vialing that into the vial and making the drug product. So just again, a little bit more of a zoom in on what chromatography is for those who are not familiar. It's really taking, the first step here that we typically use is affinity chromatography, where we take where AAV is in a combination of cellular debris. It could, proteins, lipids, other things that we are not part of the process that we want, and we run it over this column shown here, and it has affinity, so the AAV binds to the resins. And this is actually, I should point out, this is a major improvement over the last decade that we've made within AAV manufacturing. These resins didn't exist 10 years ago for the most part, and so we had to, it wasn't as simple. I think it goes back to just overall process improvements as we see within the field. So AAV binds to the column, and then we wash away all the cellular debris that we don't want, and then we elute, we wash off the AAV, and then we take that through a second step, typically to remove the empty particles, because what comes off the first column, it's not specific towards empty or full particles. So we want to reduce the amount of empty particles that are there, and so we take that through typically an ion exchange chromatography step, where again, the AAV binds to this column, and then we wash away the empties and then elute and get the full particles that we want. So it's just a little bit of a zoom in for those who may not be familiar with what the chromatography steps are doing. All this work takes place in clean room facilities, and some of you may be aware of sort of how this works. But really, the idea is it's segregated. There's no other work that's being done with that product at that time. We very tight control of what the flows within the facility. What the personnel are typically gowned up in sterile garb to reduce just the risk of as we shed skin cells and those type things, to reduce any risk. Even though the cells within a closed system, we're trying to use as great a caution as possible to protect the products. And so, the biggest piece of this is really in providing that clean air. And so typically, I'm just zooming in on the 0.5 micron type size particles that we often look at. In a typical room, and hate to scare you right now if you're breathing, but it's hundreds of millions of particles within a square meter, cubic meter, and we really purify that air down to maybe 3,000 or so particles within a square meter when we have any open step within a cabinet. And then again, maybe not tight specifications around things that are in closed systems, but we're very, very cognizant of the air quality that we're producing this in. And importantly, we have to keep this up and operational, even if we're not producing at the time. So the facilities are run 24 hours a day, 7 days a week, 365 days a year, and so we have to monitor the air continually. And we have to verify that we're not getting what's listed here as non-viable particles. That's just dust pieces in the air, as well as viable particles. So actual microbial pieces that could be there that we really want to have none of those within the facility. So the facilities and what we're producing, and typically. You build a really large facility, and the actual workplace that we're making these vectors is a relatively small part, because you have all this support space that again is held really to those same standards. And then I just want to focus on the analytics. We're not going to go through this as well, but we really focus on the four main areas, which is the safety and quality, the overall concentration of what we have, and its ability, its potency, as well as its identity, and then overall purity. And so these assays have to be developed early, because we have to know when we're working on the process and developing the process, if we make a change, are we changing any attribute to the vector that's important for its function? And so they have to be established early. And then after we establish them, then we have to go through what we call qualification, validation, and to get approval either for starting the clinical trial or really validation for approval of the product. Hand it back to Michelle.
Really great deep dive and overview into the different choices and considerations. And so with this slide, really the punchline here is to say that as I mentioned before, there are a lot of different decisions that are made over that progression through preclinical, clinical, and into commercial. And so as you make changes along that development timeline, they become much more costly to do so from both a financial as well as a time perspective. And so the mantra has really become, through those learnings, especially the past decade of AAV delivered gene therapy, that you really do need to think about what the end it looks like, what success looks like, and work backwards and prepare as early on as possible. And in doing so, you have to think about, what is the disease that you're targeting? How does that manifest, or how does that move through the population of patients that are impacted? And certainly how many of those patients are there, because to the point of scale, scale in terms of volumes, if you start out with a process that David described that is not going to get you there to be able to reach the patients, the number of patients properly, you will have to make a change, and that will cost you time, and it will cost money to make those changes. Likewise, if you change on the analytics, as David had described as well, that too is a change that will take you a few steps backwards, and therefore, again, contribute to the cost and the development time. And so these are things that I think the field really has come to understand even more so in the last handful of years. And it's important for, again, looking at the patient advocacy audience to really understand, as you're working with a partner, a developer, a sponsor, you really want to make sure that they understand these elements as well, and that you're working with them to help understand the disease and how that impacts the patient, so that as they're designing clinical trials even, that they're taking that into consideration to really set forth in the best and most proper way. So David, you can go to the next and last slide, I believe. So we didn't want to leave you with all of this complexity and to say that there's just so much challenge. Really, again, we're excited about what AAV can do, what it has been able to do, but there are many different areas that the field, that the manufacturers, et cetera, are really working to continue to innovate and iterate. So there's more science that's being created to be more effective, more efficient with the delivery, looking at those novel capsids or new capsids that David was mentioning for better targeting and ability to really deliver for each specific disease. Moving to the right, looking at manufacturing improvements, that's one of the biggest areas of opportunity that I'm personally excited about, because we now are moving into having tools that are actually specific for larger molecules, as opposed to something that's maybe a bit borrowed from previous technologies. And in addition to that, we hear a lot about capacity constraints and challenges there. I got to tell you, there's a lot more capacity that is online than ever before. And certainly as we look out on the horizon, we're excited about what else is going to be coming available and able to serve. And moving down to the lower right, I think the agencies across the globe, and I'll say particularly FDA, EMA, have done a really great job of incorporating the patient perspective, really working to understand, AAV biology themselves, and how that can be considered more of a platform approach where appropriate. They're looking at bringing forward and have brought forward other opportunities to interact with the agencies more meaningfully, more early on, to get their feedback. And as we've seen, also have provided accelerated approval pathways specifically for approaches that are looking to help those affected by a rare disease in pediatric populations as well. And then finally, looking at how we can continue to develop how we design clinical trials. That too is an area that's ripe for change. And again, that speaks to the participation of the patient advocates and making sure that there's a true understanding for the disease, how that impacts the population, as well as really what's important to that patient population and their caregivers. And so that earlier insight from the patient communities is certainly helpful, as is the openness to looking at alternative or surrogate endpoints to determine if it is really clinically beneficial or efficacious. And so with that, that is our last slide. So Laura, welcome back. And thank you for your attention. And if there are questions, we can proceed there.
Yeah, thank you very much, Dr. Dismuke and Michelle. That was a superb overview of the manufacturing process from varying angles. And I really appreciate how you simplified a very complex system. Biologically complex, chemically complex, just purely complex. So we are starting to get a number of questions in the Q&A section. A couple of them are related to standardization. So can you just talk a little bit about, given the complexities of AAV, do you think we'll get to a industry standard or a world where there is a plug and play option for AAV technologies? Is there an off-the-shelf possibility? Talk a little bit about the platform possibilities relative to AAV that are being worked on right now.
Yeah, I mean, I highlighted it a little bit in the chromatography piece. I do think, again, there's different systems and they have they're all their own strengths. I don't know that there'll be a single universal system, mainly because different systems have a benefit that it's hard to apply towards a different another system. Specifically, if you're producing a vector that if you overexpress it in the cell may hurt the cell. That's where an insect cell may be more useful, because you can express that protein inside. It may not be, it's not expressed in the insect cell because those promoters don't work. And so you don't see the sort of toxic effect on the cell if you use that system. So I don't know that we'll ever get to a single universal system. What I would say is that we'll probably align more on purification. That's what I have seen over the years is is alignment there. Again, the holy grail is around producer cell systems, but as highlighted as well, it just takes a long time to develop. And time is the enemy of the patients. And so it's not always in the best interest to take that period of time to develop. And because Michelle also alluded to, there could be changes. And if you want to make those changes at the same time. So I don't know that we get to a single universal system for AAV manufacturing, but I think we hone in a bit more on how the purification works.
And Michelle,
Yeah.
do you want to talk a little bit about the efforts that are going on out there related to the platform approach?
Yeah, what I was going to add to David's comments is that we have opportunity to help standardize kind of coming and going. What I mean by that is, looking at the starting materials, for example, plasmid DNA, where we can standardize and have those more kind of ready to ship off the shelf opportunities. That makes it that much faster and that much easier, because then you really only need to focus as much on manufacturing the transgene or the gene of interest bearing construct. What I mean by that is that standardization of maybe like a QC analytics panel, to what David was saying. If we can get tighter on that and the expectations and alignment in around the regulatory view on testing, I think that standardization is going to be helpful, because that does mean that you know what to expect kind of going in early on, that as you move and progress through that development timeline, you will have continued support and therefore eventual success, because you've utilized that standardized testing panel.
Excellent.
I think we see, so, from, again, not trying to toot my own company's horn or Michelle's, because they've obviously tried to make standardization, and we focus on that as well with some of our systems. But we've also seen on the analytics side, again, as Michelle saying, more of a standardization around using ddPCR, digital droplet PCR, as opposed to older methods that were had less reliability and had more variability in that determination. And that's been critical, really, because you have trials that are occurring between different organizations. And there could be a difference in what we're considering what the dose is. We dose on titer on that, how many genome copies do we have, and it makes it more consistent between organizations even of what's being dosed.
Excellent. There's a couple of questions related to capsids themselves. So I think what I'll do is I'll ask all three of these questions and then send it out to you both. The first question is what % of capsids do you see are successfully packaged with transgenes in typical AAV batches? The secondary part of that is it serotype specific? And then the third question is, can you repurpose capsids, or are they generally discarded?
Okay, I'm not sure if I understand the last question. Might need a little bit more understanding of what the question's meant to be. But for the first question, I had it in the table there, and is a little bit, for HEK29, it's sort of somewhat system dependent, right? And how the system itself is optimized. And so HEK293s are typically 10 to 20% full particles that are coming out. So think about that. You have 80, 90% that didn't get the genome. Now we got to get rid of something that we don't want. And so been able to optimize that to a degree in different organizations to get to 50 to 80% full particles coming out. So that makes your life easier when you get to those chromatography steps, what you're trying to enrich. If it's 90% empty and you got to get rid of all those empties, it makes your life a bit more challenging. So we definitely focus on getting as many full particles out and optimizing that step. As far as serotype dependency, as you've certainly seen that. I've seen that probably more in a producer cell systems than other systems. And it's been challenging at times where you're trying to make a certain serotype, and then for whatever reason in that system, the capsids want to exit the cell as soon as they're made before the genome gets packaged. So.
Michelle, did you have anything that you would like to add to that, or should I move on to an analytics question?
Well, I was just trying to understand that third component to the question. And I guess what I would offer is that, much like David had mentioned with regard to the different capsids and tropism, their ability to target, that you could use a flavor of capsid, if you will, for multiple types, for delivery to address multiple types of delivery. So I'm not sure if that really gets to the question, but that's kind of my, I guess, interpretation or my response.
Excellent. And before I jump into the analytics question that is next, there was a note in the chat that I think is worth bringing out to the group relative to the platform approaches that are being pushed and brought forward by the Bespoke Gene Therapy Consortium that's out there. And so that might be something, if you're interested in the platform approaches or standardization, take a look at what the Bespoke group is doing, and maybe there'll be some additional questions or answers that you might find there. So the analytics question that we're seeing is, what new advances have the most potential to replace older technology, and what are you both most excited about relative to that?
Yeah, so again, one of them I mentioned already, which was digital droplet PCR, which has been in the again the last decade of having that be advanced, and really a shift in the entire community and field to that method. Years ago we were making vectors, and we didn't even have qPCR, quantitative PCR. We used to have to try and hold gels up to a light to determine what was the concentration. So the field's advanced quite a ways. As far as other analytics, I think where the field is going, we live in a genomics age, and so single molecule sequencing, for example, next gen sequencing, being able to dig a bit deeper into understanding the differences there, as well as digging a little deeper into what happens to the capsid shell itself after it's made. So that's really more of a mass spectrometry approach to understanding that. And so I think there's been some alignment around that again, what those techniques are used. And, I'm very excited about the developers of those technologies, because they're continually to advance and bringing new tools to the space that makes our jobs better, so we can dig deeper into the analytics.
So David kind of took part of my answer. I'm probably most excited about our ability to sequence. So one of the most important things is identifying and knowing that what you have is what you're supposed to have, and nothing else. So the advances in technology around sequencing is really, I think, going to be game-changing in this space. Additionally, again, what David said is just time is the enemy. It's the enemy for the manufacturers, for the clients that we're delivering materials to, because they ultimately want to get this to patients. And so in looking at time, one of the other areas, you look at that analytics panel and how to truncate that, but still not give on safety. They're really working hard on rapid sterility versus a traditional, what is a 14-day test to conduct, and then all of the back end readout and review of the information that really are getting that down into a measure of single-digit days. So that's something that we're excited about as well.
Why are some manufacturing studies needed more than other times? In some cases there's one set of studies, in another case there's another set of studies. Why are there different sets of data that are required by different regulatory bodies, different countries? Can you talk a little bit about that?
Yeah, I think the general approach is the same, independent of where you're filing your trial. Yeah, I think there are obviously differences between the FDA and the EMA, and there's differences between even member states within the European Union about what sort of expectations are. But I'd say overall the overall body of the package that you're providing to them is similar. I think usually probably the differences are more around requirements, say, of specifications between one party and another, as opposed to maybe a specific study. And I think Michelle also alluded to it. It's often that we see this shift in what was done in early development, and then you get into your toxicology studies, maybe you change things a bit, and then what is done in your phase 1 or phase 2 is you shift manufacturing methods, or sometimes very drastically, sometimes minor changes. And so I think there's some differences there around sort of the expectations, around comparability. How do we know that the products are the same at the end between the different places?
Michelle, can you talk a little bit about minimum AAV testing requirements asked for by the FDA when an organization is doing an IND submission?
Well, actually, I would defer to David.
Oh, okay.
I think he's much more equipped and better experienced to answer that, though.
Yeah,
Well, hand it back to David.
Yeah, it also goes back to Michelle's slide around iteration. The agency, just like us, has learned through the years of what is important and what is necessary. And they have been, I've had a great working relationship with them because they see things that we don't see. They see across between organizations and what's being filed. And so they'll often, have suggestions about what should be done as a testing paradigm that you may not be, thinking because they've already seen something in another trial. And so, but typically, again, it goes into the 4 categories that I talked around, the safety of the vector, the overall titer and potency. So there's no set rule, but they've come back, for example, to say, because titer is such a critical issue, that you need to have a really low variability in that titer. They don't say you need to run this titering test. They don't say you need qPCR or ddPCR. What they will say is your variability needs, your percent CD needs to be less than 15%, which means you have to have really tight control over that assay. And then the same thing for your potency assay. You need to have a potency assay. They don't dictate what that's going to be, but they obviously want to move away from anything that has a lot of variability, like especially an in vivo study in an animal model, and moving that towards an in vitro potency assay that has better specifications and better titer control.
I'm going to take one more question off of the Q&A section, and then I'm going to leave us with kind of some final wise words from you both. So this is a very specific question. Is it fair to say that companies and manufacturers are moving away from HEK 293 and are trying to do more baculovirus given higher yields, or is it too soon to say that?
So again, I think it's, I think there's both are utilized. And in fact, the herpes virus system as well as producer cell systems are pretty broadly used. HEK are the most widely used that I have seen. And again, it's because of the speed and the less of the having to overall change. The baculovirus system is a great system, and it's worth the investment depending on the application. So for example, if your vector needs are very high, because it produces about 10 times more vector than other systems, or can, then it's probably it may be worth your investment in time and energy to get to those, because it gets you to a commercial scale that you could actually supply to the patient. And back to Michelle's point, you don't want to change it horses midstream if you don't have to. It could set you back. So I would say it is really dependent upon what the trial is, and what the overall dosing needs are, and if it's worth the time and investment early on. Probably what I'd say is more likely you start with transient transfection and probably move to a different system later on. If you get clinical efficacy and you know that your vector is working, that may be worth your time and effort, especially if you're looking at a large commercial need.
Yeah, Laura, what I would just offer up as well is that, you mentioned the partnership between the academic researchers, patient organizations. By and large, that's been on behalf of those living with or affected by rare disease. And so as the field continues to progress into diseases that affect a larger population of people, that's where I do think we will see more need to move into, bacula systems or something that can deliver, as David said, on a much broader, larger volume scale for not only clinical trials, but certainly, for widespread commercial use.
And to follow up on that, Michelle, is, as we see an increase in the number of patient advocacy organizations or patient foundations developing these complex gene therapies, I want you to leave us all with a couple of your wise words relative to when should these organizations start thinking about manufacturing all of this complexity. When does that have to enter into the conversation in the process? And then where are you both seeing the biggest missteps that are costing time and money, which ultimately cost lives in this process?
Okay, we're real tight on time, so I'll be very quick.
Yeah.
Would advocate that organizations really bring on scientific expertise, true expertise, to help them make these decisions early on. And it's never too early to engage with manufacturing, because so much starts in that decision-making, that discovery period of time, that can, with the right organizations and the right support, really help drive change faster on the back end. So it really is all about the decisions that you make early on. And that wraps into what my caveat would be, is that people aren't thinking about that as early on. They're just going. And I think you have this sweet spot of time to really be thoughtful in your design and looking prospectively at what your success, what you define success as, and then those decisions. But partnering with really, really strong, smart people is really, of course.
I agree with what Michelle's saying, and probably just add on to that, really also focusing on analytics, as much analytics as you can early, so that you're not surprised by some feature later on that you maybe deprioritize.
And Michelle and David, thank you so much for a fantastic presentation, demystifying how to create an AAV vector. So with that, I want to thank everyone for joining us for today's Lunch and Learn. This should be available. I think I'm going to hand it back to Allie, and she can describe how you can get access to this session on demand. But again, thank you for your time, and thanks for being here.
Thank you.
Glad to be here. Thank you.
Yes, thanks everyone for joining us today. You will receive an email if you're registered to view this on demand and share it with anyone. And it will also be available on the patient education website for use. There is no Lunch and Learn in May. We have the ASGCT annual meeting, so we hope we'll see you there. And I also want to plug a half-day workshop we're offering leading into the annual meeting on May 15th. It's what to expect as a participant in a gene therapy clinical trial. If nonprofit and volunteer patient advocates can register for free. And we have a great lineup of speakers ready to share their experience in running gene therapy clinical trials and working with the participants. We want to make sure we help prepare the community for this complex process while also managing expectations. The next Lunch and Learn will take place on June 23rd, and we'll actually be reporting highlights of some of the latest research presented at the annual meeting. So we'll be summarizing what might be of interest to the patient advocate community. So we hope you'll join us there. Thanks, everyone. Have a wonderful week.
See you in DC.
Thank you.